Hi, this is Blair. This video is a little weird. There's like a droning sound I can't get out of the background. I can't get Zoom to work for whatever reason. I can't screen record, so I am using a different function to screen record, and it will not show that the slides are changing if I go into full screen mode. So we're just going to be staying like this for right now, and hopefully that works. So sorry if that's bothersome, but today we're going to be looking at PCBs, which are an environmental toxin, and we're going to be looking specifically at their effects on BY2 cell division and mitosis. So PCBs, research has shown that it's likely that they inhibit the buildup of cyclin D, and cyclin D allows for the progression from G1 phase into S phase. So this experiment is suggesting that if we expose BY2 cells to PCB, then A, we're not going to see DNA replicating and being synthesized, and we're also going to see that cell division is likely going to halt completely. So PCBs are polychlorinated biphenyls, which are man-made chemicals. They were made mostly for electrical equipment, but you can still find them in motor oil, adhesives, cable insulation, etc. However, shortly after being implemented, they were banned in 1979 because they discovered a lot of risks to the people that worked with this equipment that had PCBs incorporated into them. However, the danger of PCBs, even though they were banned 41 years ago, is that they don't break down easily, and they're still cycling through our environment, and they're still posing a danger to people that may happen to come in contact with them. And the EPA has a whole section on their website about PCBs and how to locate them and how to dispose of them adequately. So it's definitely still something that, even though they were banned 41 years ago, it's still super important that we're thinking about them. So as we just said, research has found a pretty likely connection between PCBs and an inability of cyclin D to build up. And we might recognize this graph right here from lecture. And it talks about the progression through cell cycle. And we see cyclin D right here is highlighted. And you can see that it's this little gray strip right here. And it's showing that cyclin D is allowing for the progression of G1 into S. And it's being able to overcome this restriction point if you have enough cyclin D to do that. And right up here on the right is the cell cycle stage diagram that we made like 3 weeks ago, but specifically paying attention to G1 and S right here. We're noticing that as we go from G1 into S, we're seeing DNA being synthesized. And as cyclin D supports this progression from G1 into S, then PCB should stop this progression if it's not allowing for any buildup of cyclin D to support that progression. So this experiment is analyzing the continued effects on PCBs. It's showing that they're really important, even though they're banned 41 years ago. But it's starting with BY2 cells that are entering the growth phase, and they're going to be treated with PCBs from a PCB kit that you can get off of Sigma Aldrich, and that goes for $92.60. And we're going to be taking 5 milliliters of BY2 cells, and we're going to be treating them with 200 microliters of PCB, and then we're going to be placing them back into an incubator at 28 degrees Celsius, 120 RPM, and we're going to let that sit for an hour. And just noted right here, like every other experiment, it's going to have a control that is going to be completed in the same way. It's going to go in the same incubator for the same amount of time, but it's not going to be treated with that 200 microliters of PCB that the first sample was. Both samples are going to be treated after they come out of the incubator. They're going to be treated with Farmer's Mixed and then stained with acetoaresin, which we talked about 3 weeks ago, and you can find in the lab manual. Both samples are then going to be analyzed and compared in order to determine if there were any noticeable effects on the cell cycle due to the PCB that we introduced. So it's a little hard to see here, but you can see this graph here shows the personal and partner data that I got and my partner got, and then the whole class data about the relative length of stages in BY2 mitosis. And you can see that the longest stage in both graphs, in the personal and partner data and the whole graph, the whole class data, is that interphase is the longest phase, and you find the most cells in interphase when you're looking at them. And that's followed by prophase, then metaphase, anaphase, and then finally telophase. Despite agreeing that this proportion is correct, when we're looking at this PCB contamination experiment, we're not going to be seeing those same levels that we see in both of these graphs. Down here, we're going to be seeing that likely we should see interphase in 100% of these cells if the PCB is reaching and affecting every single cell. So this data suggests if we see that significantly almost all of the cells are in interphase, then PCB does have a significant effect on the cell cycle, and it is halting it effectively. The cell cycle is being disrupted by this PCB ability that stops the buildup of cyclin D, and this result was supported by this finding that we talked about on the very first slide. But specifically in this finding, in this research study, they gave us this specific graph that you can see that it's a little small, but you can see that G1 is listed right there, S phase is listed right there, and then G2 into mitosis is listed right there. So you have these three separate areas, and it's labeling the DNA content of each stage here, and then it's adding in sequences of 5, it's adding microliters of PCB, and you're seeing that as you add more and more PCB, you're seeing the DNA content, specifically of S phase and G2 into mitosis. You're seeing their DNA content is going down and down the more you add, the more PCB you add. So this is suggesting that an increased exposure to PCBs is actually lowering the DNA content after G1, this first spike, which is what we initially predicted. And these results are obviously important. They show an extreme effect of PCBs, and they're obviously still important today because they're still polluting our environment. A great follow-up is to determine how PCBs are actually causing this inability of cells to build up cyclin D, and what conditions we can put PCB in order to stop this ability and stop the continuation of cell division halting. And then an obvious limitation is that this was only conducted on plant cells. And even though both plant cells and human cells utilize cyclin D, we can't conduct this on human cells, despite the fact that a lot of the uproar about PCBs are about how they affect human cells. Because obviously testing on live human cells are considered normally scientifically immoral in most circumstances. So I hope, despite all the weird screen recording issues, that you enjoyed that. So thank you for watching.